This computer. So, if you are here with me, you have a question to ask, you can just raise your hand, or maybe you send a message in the chat. I'll be able to see that and I'll respond to your questions. Um, Welcome once again. I know this lecture was something that I had delivered before, but I felt like the information was disjointed then, and I didn't explain the concepts very well. So I'm trying to do a makeup lecture on that. You can use both of them if you want, or this one will be the one that you will most likely have a lot of information from. So today we'll be talking about DNA, which is, the genetic material. We'll try to explain how DNA, once again, was proven to be the genetic material in the biological sciences or also in living things. Because if you remember well, like we explained, there was a lot of controversies when it comes to selecting what is really the genetic material. Was it DNA or was it, protein? So, and that was largely in part because protein was a lot bigger and protein had so many molecules compared to DNA. So DNA just have five nucleotides, four nucleotides, five when you combine both DNA and RNA, whereas proteins, we have over 100 plus proteins, but the ones that make up, we have over 100 plus amino acids, but the ones that make up the proteins, the functional proteins in our body are 20, right? So they said 20 compared to 4, 20 will always be more. So that is why a lot of scientists felt that protein was the genetic material rather than DNA. Okay, so the first thing that was done to prove that DNA was, the first thing that happened was this particular experiment, right? There was an experiment that was carried on by a scientist called Frederick Griffith, right? Griffith was a scientist who tried to study let me try to get my presenter view right here. No, what is going on? This thing too hard to do. Okay, so there was a scientist called Frederick Griffith, right, in 1923, who studied the Staphylococcus pneumonia bacteria. So what he did was that he isolated a pus from someone, from an infected person. So what he observed was like after what Mr. Griffith did was that he isolated a pus from an infected person, right? After isolating the pus, he wanted to investigate what was causing the infection in that particular individual. So he took out the pus and then carried on some kind of, purification process. After the purification, he discovered these two things that are associated with the bacteria, the Staphylococcus bacteria. So this is the bacteria, but the bacteria had two strains, right? It had a rough strain as well as the smooth strain. So this is a typical image of how this looks like under a microscope. So this is the rough strain. If you remember in the previous lecture, I told you the rough strain had like, this mesh-like or rough outer portion of it. And these are the strings, that are with that rough strain. These are the ones. And then they also have the smooth strain. And the smooth strain, if you look at it physically, you see they are very smooth. They don't look that kind of rough-like as compared to that this other one. And one thing we got to learn in this was that a rough strain was the one that was not lethal. I mean, it's not dangerous, whereas the smooth strain was the one that was more dangerous. So everything about understanding as to whether DNA was a genetic material began right here. So this is where everything started. When the infection happened, they realized that the wound started expanding. And all of us have the same thing. Whenever we sustain injury, or you have a wound, or you have an infection, you realize that the pus tend to expand and damage more tissues, and then the wound get bigger. So, but for this one, it was a Staphylococcus pneumonia bacteria that we isolated it from. It is not always the case in all infections, but all infections have the infectious agent, what infect you, always has a certain form of a DNA. So for them, they wanted to investigate. This was the beginning of it, right? Now, as time goes, so what Mr. Griffin did, he isolated both the rough and smooth strain, but Mr. Griffith didn't know, and scientists then didn't know which one was the dangerous one, which one was the lethal one, which one kills. So, but they knew there were two things there. And now understanding which one kills, it means if for you to carry on some kind of information, for you to carry on some kind of information as to whether you are a killer or not, it means you need to have some sort of instruction. And if you remember in our introduction of genetics, we say the genes are carriers of instructions. They carry out instructions or information, right? They do that, right? So it means if this guy, the smooth guy, is a killer, means he have certain type of gene in him. He carries a certain form of instruction, and that instruction is to kill. All right. So now, building up from that experiment that was done, from that isolation that was done by Mr. Griffith, other scientists, you might remember, the first experiment was done in 1929. After that, the next experiment was done in 1944. I mean, it started in 1932 up to 1944, this experiment was going on. This one to prove that the S strain is the deadly strain rather than the rough strain or the R strain. So for that, these three scientists called Avery, Colin MacLeod, and Maclean McCarthy, these three scientists were the ones that spent their time trying to investigate this. And all of this work was done in the lab of Avery, Dr. Avery. So all of this work was done in their lab. And so this experiment was called the DNA as the active agent of transformation. So what kind of transformation is taken on in that particular smooth and rough strain? What was causing it? What was causing the tissue damage? What was causing that injury to extend? What was causing people to die from that pneumonia infection, right? So here now, the experiment that they did was they isolated the smooth strain from the bacteria. So here you can see this is a bacteria, the Staphylococcus bacteria. They isolated the smooth strain from in this bacteria right here, and then injected it into a mice. And what they realized was the mouse died, right? The mouse died. And when the mouse died, they realized, okay, this has certain kind of information in it. And that information means kill. That is the S. And the next thing they did was they took the S strain, they mutated it to R, right? This, we already talked about mutation, and it's something that we also talk about a lot in this course when it comes to mutation. They change certain form of, small form of mutation in this. So a mutation in this S strain is what gives rise to the R strain. So the R strain is not there by itself, but it's a form of natural mutation that happens. So the R strain mutated, or the S strain mutated to the R strain, which makes it non-lethal. So it doesn't harm anymore. It's like when you take a snake and remove all of its teeth, you remove everything like, the venom from in it, it becomes harmless, right? So now this R strain was injected into the mice and it didn't die. So what does that tell us? It tells us that the S contains certain genetic instruction that says kill. But then when it mutates, when mutation happens, that genetic instruction that says kill no longer functions, right? It's no longer active. So now that when a mutation happens also, it changes from S to R, right? So that is clear. Now let's come to C. We have C, and C, we took the same S strain again, the lethal strain. We took it and heat killed it. We put it under heat. And for those of you who work in the lab, that most of the time we carry on, sterilization, right? We warm things. We tend to, there are two things we do to kill bacteria. We either disinfect or we sterilize. So sterilization is using heat to kill microorganisms that we cannot see. And it's mostly used in the microbiology labs. In the virology labs, we mostly disinfect, right? So here we have this S strain. They use heat on it intensely, and then they kill it. It didn't change to R. This is not a mutation, right? It still remains an S, but this time around it's an S that was heat killed. So if you look here, you see the particles are damaged. And they took it and injected it into the mice, and the mice lived. So what does that tell us? It tells us that one, heat can damage DNA. Heat can damage any genetic information, right? Two is that when this undergoes heat at certain condition, it dies. It no longer functions the way it is supposed to. If you just join me, please mute yourself and then, stop your video. Other than that, you hide the slide. Okay, so what does that indicate is that mute yourself, please. Mute yourself, please. Hello. Okay. So what does this tell us? Again, is that once you hit kill something, it no longer kills, it no longer, causes that same damage. Now, here is the fun part. I say, okay, now we know S strain kills. R strain is when the S strain mutate, it becomes R. When that R is injected into a mice, nothing happens. And then we have the S strain. If we take it and put it under intense heat condition, it gets deactivated. We inject it into a mice, it doesn't kill you. Then we have the S. Again, the S, we took it, we heat kill it. Remember when we heat kill it the first time, it doesn't kill, right? The mice lived. And then we took the R strain from B. So now D is a combination of both B and C. B, mutation of R, C, heat killing S, right? So in this S now, in this D, we are combining both B and C. Please try to understand this thing. This is very, very crucial for your journey in biomedical science, as well as, as a medical practitioner, or whatever it is that you are practicing. It's very, very important for you to remember. Then we have the D. So it's a combination of both R and that of the S that is heat killed. So they took the S, heat killed it. We have this one. And remember, when it's heat killed, it doesn't kill, right? And then they took the R, the one that also doesn't kill on its own. They combined both into one syringe and injected the mice. Once they injected the mice, they found that the mice died. Individually, these two things doesn't kill, but when they combined, it kills. Then what happened? That was the biggest question. And if you remember, now 105, I rewarded somebody with something, right? For this. So what happened here is transformation. So that means something transformed from being a non-lethal to a lethal, right? Somebody influenced the other person between these two. So what they did was they took out the tissue, they took a sample of this mice and analyzed it. When they analyzed it, they found the S strain has been recovered. So it means, of course, this one is dead, right? This R strain is not a dead one, it's a living one, but it had a mutation. So in the presence of this dead one, this R strain was able to revert back to becoming an S strain. Of course, that one was being heat killed. So that was a mystery, right? Everyone was fascinated by this. It was a mystery, right? So how did this happen? So that was the theory of transformation, right? Now, the next thing was to try to investigate what was the transforming principle. They realized that, okay, here, first, we took the infection. We realized I had both S and smooth, but we didn't know what was causing death, right? And then here we have investigated, we know the S strain is the most lethal strain, is the one that killed. Now we realize that both S and rough, one can kill, one cannot. And if we combine the two of them, one convert the other. So now the scientists became more puzzled now. They say, okay, now what is the transforming principle? What is going on? What is causing this? That became the next area of investigation. All right. So remember, if you have any question at any point, you can stop me and you can just raise your hand or make a comment in the chat room. I'm going to respond to your questions. Okay. Now, another investigation was carried on, continuing from our last one. So if you look here, you see the pictures are still the same, right? So I try to make this note as simple as I can so that you'll be able to, so that you'll be able to not to get so much difficulties in trying to understand it. So DNA as the active agent of transformation, that was the next thing. They realized that, yes, we know something is going on here. How do we know what is going on? What is causing this transformation? That became the new phenomenon, right? That became the new fascination. Here, cut all matter short, they say DNA, the transforming principle is DNA. But now let's go through the experiment. Let's look at A, first of all. We have A right here. We have the living R form. You remember, living R doesn't kill. And then we have the heat-killed S component in the medium. So we took the two of them, we combined them. If you combine these two, the living R form that doesn't kill, the S form that doesn't kill, that is the S form that is heat-killed doesn't kill. If you combine the two of them in a tube, you leave them over time, what you realize that you recover a living S form. This time around, they're not doing it in the mice. They're doing it in a test tube. You see behind here, they did it in the mice. The mice died. But they say, okay, now let's recover the two of them. We don't need to put it in the mice anymore. Let's put it in a test tube and see. They put it in a test tube. They allow it for some time. And after that, they took out a sample of this and analyzed it. And they recovered the S form, which means between these two, one of them is influencing another. And we know that it is the R that is getting converted back to S in the presence of this heat-killed one. So then here is, in this B, is just a form of pellet, right? After centrifugation, after thorough centrifugation, this is the pellet they had. And now, what do we need to do? And we know in this particular stuff, in this particular composition right here, this substance that we have here, we have so many things in that. Remember, in our blood, we have so many things, right? So what do we need to do now is to carry on an elimination principle. So it is agreed that in this particular substance, there is protein, there's RNA, there is DNA, and there are other physical and chemical elements that are present there as well, like carbohydrates, and other things. All of them are present. Now that we have all of these candidates, we need to know who is the culprit, who is the bad guy. Is it protein? Is it RNA that is carrying the transformation? Is it DNA? Or is it other fats molecules such as lipids? Or is it, other carbohydrates molecules that are causing this problem? To do that, we purified the transforming principle. We took this one, we purified it, right? We put it right here. Now, we have the first thing that it did was, because like I told you, everyone believed that protein was the guy who caused this transformation. Protein is the thing, nothing else. And that was because protein was a lot bigger than DNA. Two, proteins have more building blocks compared to DNA, right? Protein have 20, right? Amino acids that make up a protein. And then DNA only have 4 nucleotides. So the first thing all of them rushed and they started with DNA, with protein. So here is a protease. A protease is an enzyme that degrades protein. All right. So if you take this sample, you put the enzyme protease in the sample, all the protein that is there will get away. All of it will get destroyed. So now, after adding proteins to this particular tube, the protein destroyed. What you see here, protein is destroyed. And what they took the sample of that after getting rid of the protein in it, they introduced it into our cells. that is the R cells, which is this rough cells, right? They are no longer treating it in the mice. So they took a tube right here, a tube containing only the R cells, and introduced it into that. And then the S cells transformation took place. They recover the same S cells. They say, what is going on? They realize that, well, protein is not the one that carried the information to transform these R cells into S cells, right? Now, they say, okay, protein is eliminated. Protein is not the one. It became more mysterious now. The next thing they produce is RNase. And RNase is also an enzyme, right, that has the ability to destroy all RNA that is present within a sample. So they added RNase again in the sample containing this R form, right? This R form, we know that R form is the one that converted to S form. So everything we do, we put it in a tube that contains only the R form. So we put the RNase there. RNase were destroyed. We introduce it into the R cells tube, and we still recover the S. We say, wow, something seriously happening here. So we eliminated RNase. We say, no, RNase is not the one that's carrying this particular process. Now we come to the DNA. And DNase, anytime you see is there, it means it's an enzyme, right? So we have DNase. DNase also have the ability to destroy DNA. And once introduced into the tube containing the R cells, there were no R cells. There were no S cells. You see here, S cells, but here R cells were recovered. The same R cells that were in that particular tube were the ones that were recovered. What happened was no transformation took place. That is because DNA that is responsible to make transformation to happen was destroyed. The guy that was supposed to make it happen was already being killed, so it didn't happen. But here, you kill RNase, it happened. You kill protease, it happened. But you kill DNase, it doesn't happen. So DNA is the one that carries out the information. That wasn't enough. They eliminated, to complete the idea, right? To make sure that nothing but DNA is the one that carry out this transformation. They also carry proper ultracentrifugation. Ultracentrifugation is like a kind of centrifugation that's even more than our normal centrifugation that we see. So they eliminated all the fats, introduced the R cells, and transformation took place. So far, so good, only DNA did not. And physical and chemical analysis was also done that indicates predominance DNA. So what they did, they took this sample, they carried out some kind of physical and chemical analysis, they realized that DNA was more present in this sample than any other thing, any of these. So then the beginning of the acceptance that DNA was the transforming principle started. Everyone started to acknowledge it, but still people were arguing. So we'll have to go through some other, experimentation to realize that, right? So let's move. Now, to prove that DNA was actually the transforming principle, again, there were other experiments that were carried out in living organisms, besides the rat, right, besides the mouse. So for this, they used this bacteriophage T4. And you see a bacteriophage, we talked about this last time, we say it has a head. DNA is inside the head, right? And then they have this trunk, right? A contractile tail that has the ability to contract, it can move up and down. They have the tail fibers, the tail, and so on and so forth. So here is a electron micrograph, right? If you visualize this bacteriophage in a sample. So all this mesh-like structure here, these lines you see, is a DNA of this bacteriophage. So when it gets into the sample, it releases its DNA. So that is what it does. So this is a 200 nanometer visualization of a bacteriophage, the size of this bacteriophage, right? Okay, so this is what we're going to use. So what scientists does is that they have the ability to open up this head, put DNA of interest in it, or dye that DNA and put it right in this head and allow this bacteriophage to go and infect a bacteria, and then we will know what happens. Now, before going to that, let's look at the life cycle of the bacteriophage. How does it operate? How does the bacteriophage operate? First, it is called a bacteriophage because it is a kind of a virus. Remember, a bacteriophage is not a bacteria. It is a virus. It is a kind of a virus that eat bacteria, that infect bacteria. right? So that's why they call it a bacteriophage. It's a kind of one that destroys bacteria. That's why they name it bacteriophage. So how do they operate? One, they infect the bacteria, right? They come and sit on the bacteria at time zero. Here's a programmed bacteriophage that come and dock on the bacteria. This is a bacterial DNA. So bacteria have circular DNA. We'll talk about, the types of DNA. So we have the bacteriophage sitting on the bacteria and releasing its DNA into the bacteria. So this is what it does. And when this happens at time 2 minutes, we already have DNA synthesis phase specific mRNA begins. So now once the DNA is released, you have special mRNA in that bacteria come and synthesize that DNA. And then after in step 3, you have replication of the phage DNA begins. Host DNA has been degraded by phage encoded nuclease. What happened is that after this bacteriophage enters, it releases its own DNA. It has the ability to destroy the bacteria's own DNA and use that to build itself. It destroys the bacterial DNA. That's so cruel, right? But it wants to survive too. That's the only way if a bacteriophage is to reproduce. If he is to born his own children, he can only do that in a bacteria. He can't do it on his own. So you realize at the end of the day, you have a lot of children of his own, right? So at time 14 minutes, see how short it is? You see how the whole short, how fast? That's why when you are infected with a bacteria, it will tend to replicate very fast. This is the timeline. So it's not the bacteriophage that has a time of replication, it's the time that it takes a bacteria to replicate that it uses, right? So it's using the bacteria. Normally, this bacteria is just replicating. This is the timeline for its bacteria to replicate on its own. So, but the phage uses this as its own advantage to replicate itself as well. So here we have the T14. It's there after we know that we have this mRNA, we have this mRNAs, we have different types of ribosomes that are there to synthesize DNA, synthesize needed materials, right? So at T14 or T time 14 minutes, DNA filled heads. Now, before we never had head, so they created the heads, they created a trunk, they created a tail, right? And then they have the DNA. Because this DNA, these are all the essential materials that have been created at this point. We have the tail fiber, right here. We have the tail fiber, we have the trunk, we have the head, we have the DNA. These are the 4 components that make up a bacteriophage. 1, 2, 3, and 4. Okay. So now that these are produced, the next thing is the assembly process. Just like making a car. So the assembly process now is to make sure the tails and the legs, everything joined together, right? The first intact phage particles are assembled. After the assembly, all of them now have to burst out of this bacteria. By bursting out into the bacteria now, so they say at T25, in 25 minutes, the host bacteria is lysed, releasing about 300 progenophages. About 300 of these guys have been produced in less than 25 minutes or in about 25 minutes. So this is a life cycle of a bacteriophage. This is what happens. So now scientists took advantage of this. But one thing I want to start telling you from here is that when this bacteriophage was releasing its DNA, only the DNA went in. You see here, you see the head, the tail, the legs, none of it went inside, right? So this is essential. So you should realize that this bacteria only needs its DNA to develop all the other parts that it needs. It doesn't need the proteins, it didn't need any other part of that. Only the DNA it needed. So all of the information that is needed to carry out this infection and reproduction is encoded in the DNA. It don't need the legs. Is that clear? Fine. Now let's move. Now, this is a life cycle of bacteriophage lambda. So if you remember, maybe you might have heard of, this lactic cycle and the lysogenic cycle, right? So it's just 2 different pathways of a bacterial life cycle. And then, phages actually take the lysogenic cycle. This is the normal reproductive cycle of a bacteria, and this is the one that the phage take advantage of. So we have an E. coli cell, an E. coli chromosome, which is the DNA of a bacteria. They have a circular form lambda chromosome. So the bacteria will come and join and then release its DNA. If the phage releases DNA, so this is the bacteria DNA, this is the viral DNA, this is the microbe or the E. coli DNA is a bacterial DNA. These two guys can go either ways. If they go through the lysogenic cycle, let's start with the side, the lysogenic cycle, the viral DNA will integrate into the bacterial DNA. Two of them will join together, and then they will produce two total bacteria. Scientists usually use this process as well when it comes to biotechnology. You know, when we're dealing with large pieces of DNA, we want to do cloning or genetic modification, we do this, right? This is done. So you just take a piece of DNA, put it right in here. Bacteria have the ability to incorporate any DNA that is available within that domain. This is called site-specific recombination, all right, to recombine. Now let's come to the lytic cycle. Lytic means pause apart, damage. Lysogenic means join and reproduce. So many viral chromosomes have been produced on its own in the lytic cycle. And then what happened, these viral chromosomes they push this particular bacterial DNA aside and allow themselves to take all of these enzymes that the bacteria normally use to reproduce itself and use it for themselves. And then at the end of the day, see cell lysis. The lysis now, which is the bursting, is what make it a lytic cycle. So these are the two different cycles of, bacteriophage reproduction or the E. coli cell reproduction. Now, experiment with viruses provide convincing evidence that genes are made of DNA. Here again, we come to the same bacteriophage. This time around, we are dealing with the T2 bacteriophage. Remember, T2, T1, it's not a big difference, right? It's the same bacteriophage. It's just the structure of these bacteriophages are almost all entirely the same. It's just that their function sometimes differs. That's why they call them the T2, T4 bacteriophages. All right, so now let's talk about the anatomy of the bacteriophage or the physical structure of the bacteriophage. We see that it has a protein coat. So that means all that head, the encapsulated part of it is a protein. It's made up of protein. Remember, proteins are different from DNA. Protein, amino acids, DNA, nucleotides or nucleic acids. So we have the DNA in here, which is this red line. And this is the host cell wall. That means this is a bacteria. The guy will come and dock there and then release his DNA in, allowing his protein to stay out. Right? You see it? Good. Now, this is the T2 phage area, right? All right. So let's come to B. The first thing that happens is that the phage attaches to the bacterium. After the phage attaches to the bacterium, which is the host, it injects its DNA into the host cell. When that happens, remember his particles doesn't come in. If you look here, you see that he already emptying himself, right? And the phage DNA replicates, new proteins begin, and the particles assemble, the same process we just talked about, and then they burst out. Okay. Now, after that happened, people still didn't believe that protein DNA was the carrier of genetic information. They felt that it was protein. So they have this thing called the Hershey-Chase Warren blender experiment to finally agree that yes, indeed, DNA was a carrier of this genetic information. Now, what they did was that they had these two phages, T2 phage, T2 phage. They infected the E. coli and grow in 32P medium, containing medium, right? And they took the same two phage here. So in this medium, they have the 32P. 32P is a kind of a dye that has the ability to attach to DNA and only DNA. And then they have the 35S. 32P is 32 phosphorus, 35S is 35 sulfur. So these two guys, the sulfur only attaches to the protein, and then the phosphorus only attaches to DNA. So now, the phages with 32P labeled DNA, so it is the DNA, the color here is the same as here. Sorry if it doesn't look so much alike. The 32P DNA, it will come and then bind to this bacteria. When it binds to this bacteria, it releases its DNA inside, right? It releases its DNA inside. They blended briefly, when they carry out the recovery process, they realized that only the DNA was present in the bacteria, and the bacteria went through that process of reproducing a new phage. But in the 35S, phages with S level of proteins were placed on the bacteriophage. Yeah, the same process happened. It releases its DNA, but this time around, you see the protein shells are outside. You see all these purple things stayed outside, right? So they are ghosts. They don't have a soul, right? The DNA is not there. So they recovered that this guy, this particular cell, no phage grew in it. There was no phage, no bacteriophage grew. Only this one with the DNA had bacteriophages. This one didn't. So they realized that yes, DNA was actually the transforming principle, was the one that was carrying out genetic information. Now, another experiment was carried in plants. The TMV, they call it tobacco mosaic virus, which is a genetic material. Their genetic material is RNA. So what you realize here, if you look at the plant, the tobacco plant, you usually get infected with viruses. TMV is a plant virus. It affects only plants. So what they did in the TMV, so this is TMV type B and TMV type A. They took the TMV type A, they degraded it, they took out the RNA, right? And then they took TMV type A, degraded it in RNA A, which is a protein A and protein B. They remove the one they took. For this guy, they took only the protein that covered him from outside. That is the covering. They brought it here. For this guy, they took out the RNA from inside and put it here. So you look at the protein and the RNA. Took this guy, only the shell, the coverings. Remember, I told you the covering is mostly the protein. The DNA is always inside. The DNA cannot expose itself, just like we humans, right? Just like we humans, almost all of our body entirely is just outer portion is protein, the DNA is inside. The reconstituted mixed viruses infected the tobacco leaf. After they took this, they infected the tobacco leaf. And then when they analyze it, they realize that the protein, you see this blue right here is the RNA, right? They recover everything to be blue. That means the RNA had the ability to transform this particular yellow one to be blue because the RNA was the one, the RNA and DNA both carry genetic information, was the one who had the ability to do this. So the genetic material of tobacco mosaic virus is RNA, not protein. TMV contains no DNA. It is composed of just RNA and protein, and that's okay. So you have the DNA structure and RNA, DNA and RNA structures, right? So now that we've already we've concluded on that, now we know the structure. Now we know how DNA was proven to be the genetic material, the carrier of genetic information, right? And the next thing we want to talk about now is the structure, DNA and RNA structures. I have a meaning. So for that, I'll leave it for our next lecture. Very sorry. Let me see the slides. Yeah, so require a lot of explanation. Okay, I can get late for that meeting. So far so good. If you are in this meeting, do you have any question? Please come on, Rufus and Tusabi. Hello? Unmute yourself if you are not sleeping already. Okay, so let me just try to run through this one quickly and we'll go to the okay. Right. Let's go. So now that we know DNA is the the one that carries genetic information, the next thing we want to do is try to understand what are the structures of DNA, which became the next area of infatuation. Everyone was now okay, now we agree. Scientific community, some people find it difficult to agree, but yeah, I don't know, it happens a lot in the science domain. People are more ego, people have a lot of egos in science, right? So everyone agreed eventually. So the nature of the chemical subunits in DNA and RNA. Nucleic acids are composed of repeating subunits called nucleotides. Each nucleotide is composed of 3 units. Now, all DNA have 4 components or 3 major components. One, they have a phosphate group, which is right here, right? 2, they have a 5-carbon sugar or a pentose. Now, this 5-carbon sugar between DNA and RNA is divided. It has a slight difference. So for in RNAs, which is called a ribose, we have 1, 2, 3, 4, 5 carbons. But in all of the carbons, you have hydroxyl groups. In carbon 1, you have the OH, carbon 2 OH, carbon 3 OH, carbon 4, we have this, methyl group or methoxy group. And then in DNA, which is called deoxyribose rather than ribose, you see the name here is ribose, here is deoxyribose, which means at 2, you see 2 deoxyribose. That indicates at carbon number 2, you see carbon number 2, we don't have an O here, an O has been removed. In comparison to RNA, you see O is there, but DNA, there is no O. So they say no hydroxyl group. Instead, we have a hydrogen group. So that is what sets the main difference in terms of structures, in terms of chemical composition. In the chemical composition of DNA and RNA, this is the main difference. All right? So there's no O here, but here we have O. In RNA, we have the O. In DNA, we have no O. Besides these two, the ribose, the pentoses are the same. There is no difference. Let's go to the next one. So we say, yes, they should have a phosphate group. They should have a 5-carbon sugar. And then these are the subunits, a cyclic nitrogen-containing bases. We have the uracil, the cytosine, and thymine, all right, which are the purines. And then we have adenine and guanine, which are the pyrimidines. Pyrimidines are the cytosine, uracil, and thymine, and the purines are the adenine and guanine. So here you can see RNA, in RNA, only with rare exceptions, right? You have uracil. All the time, uracil is in RNA. In both RNA and DNA, we have cytosine. In DNA only, with rare exceptions, we have thymine. But in adenine and guanine are in both DNA and RNA. Okay? But uracil is only present in RNA, and then thymine is only present in DNA. But it is not a holy grail. It is not a holy word. They said with rare exceptions, which means sometimes mutations can occur. During the times of mutation, you see where thymine is, where uracil is supposed to be in an RNA, you find thymine, that's a mutation. Okay? So these are the chemical compositions of DNA and RNA. The nature of, the same thing, right? So there are other ways they can be called, the pyrimidine nucleotides and the purine nucleotides, right? Deoxythymidine monophosphate or DTMP, deoxycytidine monophosphate or DCMP, deoxyadenosine monophosphate, DAMP, and deoxyguanosine monophosphate, DMP. So let me just email here that I'll be late for, see here, please, sorry. Let's see. Sorry, guys. Maybe it's not a good thing. I'm meeting my supervisor today and I'm late. So it's not a good impression for me. So we have this, right? The pyrimidine nucleotides and the purine nucleotides. You just look at our structures and so on and so forth. So they call them so if they are present in the DNA, we call them deoxy, thymidine monophosphate. That means the carbon number 2, there's nothing there. That's why, that is the reason for all this deoxy, all right? Because carbon number 2, nothing is there. But then if it's in RNA, we just call them thymidine monophosphate, adenosine monophosphate. But if it's in DNA, we call them deoxyguanosine, deoxyadenosine, deoxycytidine, deoxythymidine. All right. So the structure of the DNA was deciphered or discovered by these 4 individuals. All right. They have Morris Wilkins. Okay, right here. They have Francis Crick. this individual right here. This is Morris Wilkins, right? This is Rosalind Franklin, the lady with the X-ray diffraction, and this is James Watson. All right, these are the 4 individuals that contributed to the discovery of the structure of DNA. So right here is a single-stranded DNA that you'll see. You have guanine, cytosine, thymine, and adenine. Why is it a DNA, not an RNA? Of course, it's a single-stranded. Well, it is a DNA, not an RNA, because it has a thymine and not a uracil, right? We say RNAs have uracil and DNAs have thymine. Sorry for the noise. So after we discover the structure of DNA and the pairing of thymine and adenine, what we realize here is that again, adenine always pairs with thymine and then cytosine and guanine, right? So this was discovered by a guy called Erwin Chargaff, also called the Chargaff's rule. What Chargaff discovered was the possibility or the probability of thymine pairing with, the probability of adenine pairing with thymine was much higher than adenine pairing with guanine. So you see the molar ratios here. The higher, the better. So adenine and thymine have 1.8, but adenine and guanine has 0.9. And guanine and cytosine has 1.88, whereas thymine and cytosine had 1.3. So the higher, the better. So the higher the number, the higher probability that these 2 people can pair together, right? So then you can see the rest of the organisms. There are different types of organisms. And we are here, Homo sapiens, right? These are our ratios and other different organisms. So this is the structure of DNA. It is a double helix, right? This is this diffractions here. This is a picture that was taken by the only female you saw in there. It is called photo 52. It shows the X-ray diffraction, how the strand separate itself. So DNA have both the 3' and the 5' end. And here it's called the major groove, where you have 3.4 nanometer is a major groove, 0.34 nanometer is the minor groove. So we have this, DNA going into spiral form. So here we can also see guanine and cytosine, cytosine, guanine, thymine, and adenine, the pairing. And one thing I want you to remember is that wherever there is guanine and cytosine, there are 3 hydrogen bonds. Whenever there are thymine and adenine, there are only 2 hydrogen bonds. We talked about that in our previous lecture, and we're going to see it right here. So let's take a closer look at the structure of the DNA. We see that whenever there's an adenine and thymine, they are anti-parallel. And also DNA will tell you that they are anti-parallel. Anti-parallel means one strand is going down, the other is going up. If you look at these arrows right here. So a DNA is synthesized from the 5' to 3'. Okay. So if the synthesis is taking place, it will come from the 5' to 3'. So here we have now one strand is going down, the other one is going up, which makes it anti-parallel. So here we have adenine and thymine, 2 hydrogen bonds, adenine and thymine, thymine and adenine. So here we have 2 hydrogen bonds that are connecting. And then we have guanine and cytosine, we have 1, 2, 3 bonds. So this is the structure of DNA, all right? And this is also a space-filled model. A space-filled model is the way in which, scientists try to talk about for internet too, right? So about the DNA. Okay. So this is the structure of DNA. I know we talked about this. So our lectures ends here for today. And the next time in our lecture, what we'll try to do is to talk about, how DNA replicates. So that's the next thing we'll try to talk about, because that's the essential things. Or maybe we'll try to talk about the bacterial DNA and different types of other DNAs that are there. The circular DNA, I know, please also try to read the notes as much as you can, because that is very important. And in our next lecture, we'll try to talk about, the different structures of DNA. If you have any questions, you can ask me now. And then if not, we'll just try to end the lecture. Thank you. So are there any questions? Okay. Please unmute yourself if you have a question. If you don't, then we can say goodbye. Okay. All right. Since there are no questions, I'd like to end the recording right here. Then I'll try to share the recording with you as well for, future records maybe.